So, title of my presentation is Bioprospecting of Marine Fungi. And we can start with first one, that is, what is bioprospecting? What do you mean by bioprospecting? So bioprospecting means a systematic and organized search for useful products derived from bioresources. Maybe these bioresources are microorganisms, animals, plants, whatever, but development of a product for commercialization uh, using bioresources is bioprospecting. So in today's talk, we will be focusing more about healthcare products. So I'm really fortunate that Dr. Sarma Garu has already presented about the marine fungi. Actually, he is my good friend, and I had visited him in Hong Kong, if he remembers. And he's really a great mycologist, and we worked together in NIO for some time. So here in this presentation, I'll be giving more focus on bioprospecting part, and at the end, I can show how these marine fungi are useful or we are using for bioprospecting purpose. So bioprospecting completely focusing in this presentation is on healthcare products, that is drugs. So if you visit any medical store, any pharmacy, you can see a lot of medicines are already available.
Sorry,
Then question is why we need more drugs.
sorry,
So
Dr.
these are the
Thakur,
reasons for that.
Dr. Thakur, sorry to interrupt. Your slides are not in the full presentation mode. Either you go press F5 or use the PDF file.
Still, you cannot see full presentation
No,
mode
no, Dr. Thakur, no. Just
because I can see your full presentation mode.
yes, you can, but there's a press F5
Yes.
or use the PDF copy if you have a PDF copy.
Now you can check.
No, it is not in the full slide mode. Just save it as a PDF file.
Okay.
File save as.
Yeah. I'm doing that. Yes.
Let me take this opportunity to request the other speakers to please be ready with a PDF copy of your presentation in case this kind of technical issues pops up. Thank you so much.
Can you see now?
Yeah, yes. No, then you go to the full presentation mode. There must be some. No. Last at the end, no. There is an arrow is there, no. Just press on the arrow. And उधर नहीं ऊपर printer है ना ऊपर print ऊपर उसके ऊपर.
यहाँ पे.
I think you can use this slide. Then you can use the scrolling. No problem.
Okay. Yeah. Yes. So I was talking about the need for more drugs, right? On this slide, You can see this slide.
Yes, it's visible.
Yes, it is visible, Dr. Thakur. Please go ahead. Thank you so much.
Yeah. So these are the reasons why we need more drugs. First one is emerging diseases are there. You can see recently we have gone through this COVID pandemics. Then growing antibiotic resistance. Then many drugs which are having awful side effects. And some of the existing drugs are very expensive, especially in case of cancer and some of the neurological disorders, and which are not affordable. So these are the reasons we need more drugs. And then how what are the infections, especially because if you consider marine fungi, most of the natural products developed or discovered from marine fungi or in general from fungi are used for infections. So treating infections, you can see the list of infections. There are several infections coming from right from hospital acquired infections, then ventilator acquired infections, and so on and so forth. So these are the infections, and what are the reason? And then presently, these are the antibiotics available for treating these infectious diseases or infections. Linezolid is there, gentamicin is there. Almost dozen of antibiotics are available, but day by day the antibiotic resistance is increasing, and these are the reasons. You can see this was a comparison between three cities, Delhi, Vellore, and Mumbai. And if you see, in case of Mumbai, almost 36 to 40% of prescriptions given by the doctors in Mumbai are loaded with antibiotics. In Delhi, this percentage is almost 21%, and in Vellore, 41%. So mostly there is not much awareness about using antibiotics in the clinic in the patients, and without thinking or without reason, even for simple inpatients, antibiotics are being given, and the antibiotic resistance is really increasing. That's why there is a urgent need of more and more antibiotics in this particular area. So here is the data. Just now I have explained you about especially MRSA, the methicillin resistant Staphylococcus aureus, from 29%, it was in 2000, and in 2015 it was 47%. So you can see how rapidly the antibiotic resistance is increasing in the pathogens. Then second target is a cancer, and most of the drugs which are discovered for cancer or for anti-cancer purpose, those drugs are not curing cancer, but just slowing down the growth of the cancer, or maybe some whatever the prolonging the effects of the cancer on human body. And there are different targets for checking the cancer drugs. Maybe there are anti-angiogenic drug, means the drugs or the natural products which can stop the new blood vessel formation. Generally, angiogenesis is formation of new blood vessels, and this particular thing happens when there is embryonic development, there is a wound healing, or in case of tumor. So tumor is growing, then tumor is attracting more blood vessels towards it for getting supply and the nutrients, blood supply and nutrients. So for that reason, you can use anti-angiogenic drugs, and most of the drugs, the molecules isolated from fungi or bacteria from marine sources, are useful as anti-angiogenic drugs. So there are different targets for treating cancers, and these natural products derived from the microorganisms are useful to treat cancer. But in case of the existing drugs, there is not a perfect cure for cancer. There are maybe the slow growth of the cancer or reducing the tumor, those kind of things are there. So there we need a new drugs to tackle this problem. So now coming to the natural products. So far I was talking about the bioprospecting. What is bioprospecting? Then how this bioprospecting link to the natural product drug discovery in case of healthcare products or the drugs. And if you consider the natural products are organic compounds isolated from natural sources produced by different pathways, that is a primary or secondary metabolism pathway. And nature is just inspiring us or showing us a molecule, the structure of the molecule. And then in most of the cases, because we basically our group in NIO, we are working on marine invertebrates. Earlier, our more focus on marine invertebrates like sponges, ascidians, corals, and all those. But in those cases, the drugs or the molecules which are discovered from these sources are not going ahead beyond, say, toxicity or maybe in vitro or some sort of, or some experiments related to in vivo studies, because there is a supply issue. There is not enough compound available, and every time you cannot go back to the nature and destroy the nature, or the bring the sample again and extract the molecule. So what I want to say is that the natural product, in case of natural product, the nature is just showing you the structure of the molecule and application. Then you have to find a way how we can go ahead with maybe a synthetic chemistry route or mariculture or cell culture in the lab. But in case of fungi and bacteria, this job is more easy, and that's why we are focusing nowadays more on microorganisms, because we can grow these organisms, microorganisms, in fermenters or bioreactors, where there is not an issue of getting what is that biomaterial for extracting the molecules. Okay, so nature has been a source of medicinal products since long time. almost more than 50% of marketed drugs today are derived from natural sources. These are the two best examples, and I cannot escape these examples because these are related to fungi. Both the antibiotics are discovered, that is penicillin and cephalosporin, and these are presently in market for as a drug for the treatment. Then one example I'll give about the natural product, that is Taxol, that is from Taxus breviflora. This is again for treating the ovarian breast cancer, and this is from the plant origin, plant leaves. It was so. In conclusion, if you see, in case of natural product, new drugs are needed to address the limitations of existing ones, and natural products are will be a good source, or the can give you leads for the development of new drugs. And the marine fungi are a unique resource, because here in marine fungi, I just now was mentioning about the bio material for extracting drugs. So in case of fungi, even better than bacteria, because here you are getting more biomass. Then fungi are secreting unique kind of natural products, and the even supply issue of the biomass is reduced in if you compare fungi and bacteria. So fungi is really a good source for getting natural products. So basically, in case of marine, what is it, biodiversity and bioprospecting from marine sources, you can see the ocean covers the 70% of our Earth's surface, and almost, if you consider the invertebrates, I'm talking about invertebrates because in our group we are isolating fungi not only from sediments and seawater, but also from the invertebrates. There are many invertebrates where we can find the associated fungi. And you can see the greatest biodiversity, almost 300,000 described species of plants and animals in the ocean, representing almost 34 out of 36 phyla. And these marine natural products are produced because you would think that any organism, I'm talking about the invertebrate case, say sponge, which is sitting in Goa or in Mumbai coast, why that organism should secrete anti-cancer molecule or antibiotics. So basically, the purpose of secretion of these kind of molecules, the secondary metabolites, are for their own purpose. There are ecological roles of these metabolites. For example, these metabolites are used by the organism for space competition, to acquire more space and to kill rapidly growing cells of their neighbors. Then these metabolites are useful as antibiotics from through seawater, whatever pathogens are entering in their body, they are using to treat those infections. Those are antibiotic kind of thing. Then to deter the predators, they are using these natural products, the secondary metabolites, and also to tackle problem of their the associated organisms they are using. So marine natural product is completely a separate area of research, and it was started in 1960. You can see this is the birth year of marine natural product, where there were 4 compounds which were isolated from marine tunicates that time, and that was the first time the marine natural product research started. In 70s, 80s, there was again improved techniques to isolate molecules in non-polar and polar molecules. And marine natural products are not only pharmaceuticals, but you can see that many things are there. Healthcare products, enzymes are there, dietary supplements are there, food pigments, biopolymers. There are many things comes under marine natural products. But in this particular presentation, we are focusing more about the healthcare products. So if you consider the history of marine natural products, I'm just trying to impress you why marine natural products are important and how fungi is one of the good sources for getting these marine natural products. So you can see in 1815, see, we were knowing, our ancestors were knowing about marine natural products and their applications for human benefit. You can see these examples. 1815, Abermeyer mentioned the medical application of sponge. Chinese pharmacopoeia, you can find a lot of medicines are reported from seaweed-based organisms. And in our own traditional Indian medical system, that is Ayurveda, you can find that so many marine natural products are being used. That is a Praval Pushti, that is a coral powder, Mukta, Kapardika, Shukti. All these products are being used in our Ayurveda. So there is already we have a history of using marine natural products. And here you can see even people that this picture was around 18th century, where people tried sponge juice to treat eye infection. So it means our ancestors were knowing that there is some application like antibiotic thing. Here you can see one funny picture, like in case of damaged nose or any problem related to the nose, they tried whether can we apply this sponge. You can see this is a lime wood ring. This is lime wood ring and the sponge is fitted over here. So that they tried whether we can use this to treat or to replace the nose. So maybe this was not in application, but just I'm just showing that how our ancestors or marine nature product has a history, that our ancestors were knowing since long that marine organisms are useful for treating or for the human benefit. So now, marine nature product drug discovery, if you consider, and if you consider the fungi, the nature products from fungi, you can see this is an interdisciplinary area where you need people or the experts or the researchers, right from microbiology. Like here, you can see the fungi, the isolation of fungi, then molecule isolation, that is organic chemistry. You have to extract and then isolate the molecule by using different chromatographic techniques, and then go for characterization using spectroscopic techniques. Then in vitro assays, those are in outside the animal body. Then in vivo assays in different model organisms, and then a lot of chemical synthesis you can see, and then toxicity testing is there, because there should be a safety window between efficacy of your molecule and the toxicity of the molecule. So that toxicity part is there. Then pharmacokinetic is there, means how that molecule is behaving in the body, and how that body is reacting to the molecules, that is the pharmacokinetics and pharmacodynamics. Then formulation, once the molecule is ready, then you have to think in what way I can apply that molecule, whether it will be in the form of injection or tablet or ointment, whatever. So there is a formulation, and then finally the clinical trials, and the drug is reaching in market. So this journey is not very what is that, it's a time consuming, definitely, and you need a lot of money to put here. It's very expensive. But if you discover a molecule, and then if you are, that's why there are these patent rights. If you see that patent rights are for 20 years. So company or the organization which is investing time and money, they take advantage of it for at least 20 years to recover their expenses, and those molecules through this route finally reaching to market, maybe 15 years of time, and involvement of mycologists, organic chemists, then pharmacologists, synthetic chemistry people, formulation people, and finally clinical people. So it's a really a long journey. And coming to marine fungi bioactive molecules, I have given in this table, I'll be sharing this presentation with you. You can go through the references right from 2010 till 2020, and these are the molecules, different molecules. And these are the fungi, the species which are given here in this column.
Dr. Thakur, are you there? I think we have lost him.
Yes, yes.
Let us wait for.
Cytotoxic molecules, antiviral. Okay, so I was talking.
Dr. Thakur, just settle down with the network, please. Thank you. Dr. Thakur, if you hear me, just switch off your video and then present. Ladies and gentlemen, looks like there is a technical issue. Let us wait till Dr. Thakur could fix it. Thank you.
Dr. Shnoy, I hope he is aware that he has gone offline with the session.
Rohit, I'm talking to him. Please wait.
In the meantime, I think I personally thank Dr. Shnoy and Dr. Sharma for having this seminar session because it has introduced us to the topic of marine fungi. Yes, Dr. Shnoy. Yeah.
I spoke to him. He should be back soon. Thank you.
And I think I'll just add a couple of points more. What I feel that I think it's a very nice beginning by Professor Sharma by introducing us to the biodiversity, taxonomy, and the ecology of the marine fungi. That has set the base for the today's seminar on marine fungi. And definitely, the present talk is he's online.
Dr. Thakur, can you hear us? Yeah,
Can you
just
see
go
the
to your
slide?
desktop and go to your file, please. Go to that table,
Yeah.
please, that I was mentioning. Okay. Thank you.
Yeah.
Please go ahead. Thank you so much. Yeah.
So these are the metabolites which are isolated from fungi. I already explained you. And some of the metabolites I'm showing here in this slide, which are isolated by our group in NIO. And I'll be talking more about this kojic acid, okay, which is in presently in development for application purpose. And these are the compounds which are in market or in clinical trial. So I was mentioning that there are 4 different clinical trials, and really that is a critical step of drug discovery drug discovery research. And in these clinical trials, some of the couple of molecules are in already in market, and then I was mentioning about penicillin and cephalosporin earlier, and some more are in clinical trials. So that's what I've given here. So now I told you that we are working on marine fungi not only from sediments and the water, also from the organisms. So how we do that? Here is the sponge. Mostly we are focusing on the marine invertebrate associated fungi, and then we are isolating this, getting in pure culture, and then through fermentation route we are culturing them in large quantity, and then further extraction by using organic solvents, different polar, non-polar to polar organic solvents. And once the crude extract is ready, we go for bioassays, different bioassays, antibacterial bioassay, antifungal, anti-cancer, antioxidant, and some more in collaboration with our partner institutes, partner sister CSIR labs. And then finally, the molecule which we are isolating the lead molecule by using chromatographic techniques and characterizing by spectroscopic tools. So here is the process how we do that. But this step definitely from getting fungi in pure culture from say sponge is a important step for us. And here we spend a lot of time. We try different media for that and try to get more and more new cultures from these marine invertebrates. So this is about the kojic acid. Kojic acid is a secondary metabolite isolated from the fungi. And you can see there are a lot of applications of kojic acids, right, from in functional foods and in even in cosmetics. And here is the flow chart, how we are doing that. So these different fungal cultures were available with us from marine sources, and some of them we tested in the biases. This was the culture. Aspergillus venti was isolated from the sponge, from the caulerpa, and this is a chromatographic picture, that is a TLC. And finally, we could get this molecule almost 10 gram, which is available with us, and we are doing a lot of application studies now. And this cost I have mentioned, actually, kojic acid, if you see on in literature, you can find this, but this is not a real cost. It's 8,000 rupees. Actually, you can, if you can go ahead with this natural product route, definitely you can reduce this price and get the kojic acid for various applications. Then this slide is not related to marine fungi, but related to bacteria, because I wanted to highlight our story of isolating molecules from microorganisms. And this is from the bacterium. This bacterium was Cocoria species isolated from the sponge. And you can see the complicated structure, almost 1,500 cyclic peptide. And this is really effective against hospital acquired infections. And almost we tested against 250 clinical pathogens, and in vitro and in vivo studies where we could see a really good results. And now we are further developing this molecule for filing investigational new drug application, that is IND application, so that we can enter in clinical trial, and hopefully in next 1 year it will be done from our lab. So this is our success story. And what we do in our bioorganic chemistry lab, so these are our different sections. That is a natural product chemistry, very strong group. 3 organic chemists are working for isolation of molecules and characterization. We have a small group of molecular biology where we are doing even whole genome sequencing, transcriptomic studies, metagenomic studies. Then fermentation facility we have up to 10 liter, which is for growing these cultures, especially fungi and bacteria in large scale, so that we can isolate the molecule. And some small group or the section of synthetic chemistry, because as I had mentioned, when our molecule is from say invertebrate, then we need this route to get more molecule. If it is from bacteria and fungi, we go for fermentation route. Yes, so that's all about my presentation. If you have any question, we can discuss now, or you can note down my email ID, which is given here. You can contact me. Thank you so much.
Thank you so much, Dr. Thakur, for the wonderful presentation. Ladies and gentlemen, if you have any questions, raise your hand. Mr. Mohan Kumar, please. Wait, let me unmute all, please. Okay, Mr. Mohan Kumar. Yeah.
So it was a great presentation. So I'm also a microbial biotechnologist. Basically, I work with extraction of secondary metabolites from fungi, basically from mycetes. So my question is that when it comes to a fermentation process, so how do you design a fermentation media exclusively for marine fungi? Because it requires a specially designed media along with that salt concentration and everything.
Yes.
So how do you design a particular upstream processing? Also, how do you design your downstream processing for the extraction of the metabolites?
Okay, so basically we are not working much on marine fungi fermentation scale, large scale fermentation. We are going up to 10-liter fermentation in the flask in case of fungi. Of course, in case of bacteria, we have a strategy and we have even gone for say 5,000-liter culture in fermenter by designing media. But in case of fungi, we are using routine media, the broth for culturing them in the flask up to 10-liter. So we are not using what is that used to do fungal work on large scale actually at fermentation scale where we need really apply more techniques for upstream and downstream processing.
But will that grow in that particular concentration of salt? Because regular media will
Yes,
not have much salt
actually, our
concentration.
concentration is almost we keep 25 to 30 ppt salinity in our culture media because most of our cultures are from say intertidal region where the salinity is very high also, and some of them I have shown about the kojic acid is from the Caulerpa, from algae. So these cultures are from intertidal regions or sometimes mangrove areas. If we have a culture, then definitely the salinity we keep less up to 15 to 20 ppt. But 30 ppt is generally you can consider when we are growing fungi from intertidal region.
Okay, thank you, sir.
Mr.
Okay.
Arvind.
Thanks for the opportunity, sir. So it was a very excellent talk. So my question is on when we come to know this fungi is having activity towards a particular species, and we take the extract and we will see the activity as a natural product. But the natural product, the extract, will be having a lot and millions of compounds in it. But how you find out the single compound which is showing an activity, sir? So what is the tax?
Yeah, very interesting question. That is very a tricky job to go for to isolate a lead molecule for your purpose. For example, once we extract fungi and we have say ethyl acetate extract or chloroform
Okay.
extract or methanol extract from fungi, then crude extract we check for the activity. And once the
Okay.
activity is there, then we go for bioassay guided fractionation. So every time we do bioassay, and initially we use thin layer chromatography every time to check how many compounds are visible, what should be the solvent system for to be used for the chromatography. And
Okay.
literally, maybe it's a very traditional way of isolating compounds, but we go for silica gel column or other material we fix for the column, and then we go for the column chromatography and isolate different fractions. Every fraction again we check, and this way we are following up till we get the lead molecule in that. And final stage we use, of course, UPLC and HPLC to isolate
Okay.
the compound. Suppose there are 2 or 3 compound mixture we are getting, we are going for this HPLC. But basically, so far, whatever more than 100 compounds isolated by our group in last 15 years, and most of the compounds through this route only,
Okay.
chromatography and then at the end HPLC.
Okay. So
But every time
I
we check for the activity, yes.
okay. So the second question, what is your idea on the AI tools such as Torringes, Mastro, so and so? So what is the idea of drug discovery in that part, sir?
Could you please repeat that question?
No, sir. Now, as currently, now the race of Storages or the Mastro, the AI tools being come across.
Yes,
So we
tools.
can, yeah.
Yeah.
So what is your idea on following that, the latest
I
techniques, sir?
think you should go for AI tools. It is not only for, see, for example, uh, till recently or now also we are using a lot of chemoinformatics or the modeling of the molecules. And then we are
Okay.
checking whether these molecules, without doing the bioassays in vitro or in vivo assays, you can predict that what kind of activity this molecule will have or which group we have to replace and add so that this activity will be there. So that was very much advanced, but now when you are using AI tool, the same data you can, the prediction of activity of the use of the molecule or repurposing of drugs from whatever drugs are already discovered and we want to repurpose the drugs for some other application. These AI, the tools which you have mentioned in AI are really useful.
Okay.
Of course, we are not
Thank
doing, but we have to start that. This is actually the advanced techniques and we have to use those techniques.
Okay.
Because see,
Thank you,
if you
sir.
consider marine natural product drug discovery, it's suffering.
Hmm.
Almost more than 25,000 compounds are reported, but hardly 5 molecules are in market, and say 20-22 molecules are in clinical trial. The major
Okay.
or the bottleneck is the source material or the material, the biomass. Okay.
Hmm.
So if we are using this kind of techniques, AI and say modeling, and then chemoinformatics, then definitely the success rate will increase.
Okay. Thank you, sir.
Dr. Devdatta, please.
Thanks, a very nice doctor. Actually, I have one question for you, because during the process of subculturing of marine fungi, we experience a lot of loss in the production of. I mean, there will be no stable production of secondary metabolites in due course of time, and it loses its potential, any antibacterial activity or antioxidant activity. How to tackle this?
Yeah, actually, as I told you before, these molecules are being secreted by the organism for their own purpose. When there is a need, they secrete the molecule. Most of these are secondary metabolites, which we are using as a natural product for various applications. So, in case of bacteria, now we were facing the same problem in one of our cultures, which is producing potent antibiotic. Now we are going from wild type to recombinant strain for the production. So wild type strain, definitely, sir, this is always the problem. You cannot expect, or you cannot plan, or your future investment in, say, manufacturing of this any molecules for applications based on wild type, because there is no stable production in wild type cultures. Whatever media you use, and then once you are using this culture for a long time, of course, we are doing time to time subculture, but the chances of losing that activity is very much high. But recombinant strain, if you consider, that would be the better option for this.
Thank you very much, sir. Sir, one more question. Like, when you isolate the fungi from invertebrates, so like, what is the like reproducibility of the particular strain, like which is showing which is showing better activity? You said like reproducibility of certain fungi is very important during the isolation of process, right? During the isolation process, right?
Yeah, right.
Because they are
Reproducibility,
diverse also, right?
right?
Yes, sir.
Yeah, molecule reproducibility, you're saying. Yes, there is a reproducibility, because these cultures which we have isolated from invertebrates are pure cultures identified and giving the stable, at least for a couple of years, these molecules we are constantly getting from that particular strain. Of course, if we are long run, we don't know whether this wild type culture will continue to produce the molecule, or it may stop also. But reproducibility is there, definitely is there in production of the metabolites.
Thank you very much, sir.
Dr. Sunanda Thakur, the last question.
Which extraction is method is good, sir? Non-polar, two-polar?
Yes,
Whether
basically you should start with non-polar solvents like hexane or petroleum ether. Benzene, of course, we are not using because it's carcinogenic. But hexane, petroleum ether, you can start, and slowly you can go for, say, ethyl acetate, chloroform, and then finally with methanol or water. So non-polar to polar extraction is the best method. But in case of fungi, what we found that mostly in ethyl acetate and in chloroform and methanol, we are getting good, diverse, or the metabolite diversity.
Okay, sir.
okay, thank you, sir.
Yeah, thank you very much, Dr. Thakur. I'm already having fun with the kind of presentation Dr. Sharma and you have given. I think you have set a right tone for this seminar. Thank you very much. Over to Dr. Rohit Sharma.
Thank you, Dr. Shnoy, and thank you, Professor Thakur, for an excellent talk on the biotechnological aspect and the applied aspect of the marine fungi. That is an important aspect that is there. And as you told that there is a lot of potential of these marine fungi on the antimicrobial and the anti-cancer aspects also. And thank you for answering a couple of questions, or especially on the reproducibility of the molecules, because that is an important aspect that comes when we work on the marine fungi. We know that you have personal some commitments and over the day, but still, thank you so much for taking time out for the today's seminar. Thank you so much, Professor Thakur.
Thank you, Dr. Rohit. Thank you so much. Thank you, Dr. Belle.
Thank you. We'll see you soon. Yeah, thank you.
With this, we move on to our next speaker, and as Dr. Sonoy has said, that it's the right tone set